All right, we're going to go ahead and get started on the next section. This is the phylum Arthropoda, the arthropods. This is an extremely large and very diverse group of organisms. It is made up of the arachnids, crustaceans, insects, centipedes, and millipedes. So the phylum Arthropoda, there is a subphylum. We have a few subphylums to go over. So this is kind of a breakdown of all the subphylums and classes within the phylum Arthropoda. The first subphylum is Chelicerata. In that we have the class Merostomata, horseshoe crabs, and the class Arachnida. This includes spiders, scorpions, mites, ticks, etc. Next, we have the subphylum Crustacea. We have only a single class within the subphylum Crustacea. That is the class Malacostraca. That includes crabs, crayfish, shrimps, lobsters, etc. Then we have the subphylum Myriapoda. The subphylum Myriapoda has two classes, the class Chilopoda for centipedes and Diplopoda for millipedes. Then finally, we have the subphylum Hexapoda with the class Insecta, and that includes all insects. So we do have a lot to cover within this one section, and you can just see how diverse arthropods are because we are talking about all sorts of crabs, spiders, centipedes, millipedes, and all of the insects you can imagine, all within a single phylum. So needless to say, this is an extremely large group when it comes to the animal kingdom. So this pie chart that we have here, this is all life on Earth put into a pie chart. And 75% of that, all of this blue, are arthropods. All of those are arthropods. It is the most diverse phylum in the kingdom Animalia. This is one of the reasons why I decided not to become an entomologist, because you can't study them all. So there's a lot. There's too much to fit in my brain. So I really admire all those entomologists who are capable of holding all of this knowledge. And most of those are going to be insects. So a very good proportion of those are all going to be insects in that class Insecta. So phylum Arthropoda, that kind of breaks apart to mean jointed foot. They have a hardened exoskeleton. They have a sclerin protein and a chitin base. So it's a chitin and sclerodin protein exoskeleton. They're in that molecular, in the molecular phylogeny, when we had the protostomes were further divided into those Lophophocizoans and the ectozoans. So these guys are included for our ectozoans. They molt an exoskeleton. And having an exoskeleton helps deter water loss, so they can keep a lot of their water inside of their body and not dry out. The exoskeleton also provides a good protection from predators. They have jointed appendages. This is the first time we see joints. Joints are really important because it creates different points for muscle attachment. Different points of muscle attachment means we can have more muscles, different types of muscles, big muscles, little muscles. So joints are extremely important when it comes to movement, muscle mass, and muscle use. So jointed appendages provide sites for muscle attachment. They can be stronger. More joints equals more muscles. And of course, there's all sorts of flying and crawling insects. So we also have all sorts of different types of locomotion now, much more advanced than that wiggling that I had talked about in previous lectures. And we see some specialized appendages. As we all are probably familiar with, there's a lot of different types of insects out there, let alone all those different types of arthropods. And each one of those is going to have some specialized parts. They can be specialized feeding parts. We'll talk about chelicerae and palps as we go through the lecture. They can have specialized appendages for sensing their environment. So the antennae are used to sense chemical cues. They can sense vibrations. They can sense pheromones. Lots of different ways of sensing their environment. For reproduction, we have some specialized appendages for reproduction now. And we also have some specialized appendages for defense. We have pincers, as we see in crabs and lobsters. We have stingers that we have on scorpions, and of course fangs for spiders. So some specialized defense mechanisms. All right, so big concept for arthropods is that they all exhibit different types of tagmosis. So tagmosis, arthropods fuse segments for specialized functions. So tagmosis, or tagmata, are separating the body into these different segments. And you can kind of easily tell about which segment is which. In the red, this is the head. The head is going to include sensory structures and mouth parts. In the yellow, this is the thorax. The thorax is considered the powerhouse or the muscle box of the organism, and this is where the legs are going to attach, and where, if they are winged, this is where the wings will attach and grow out of. Finally, the last part is in blue. This is our abdomen. The abdomen is going to house all of the organs, digestive and reproductive organs. And depending on which subphyla and sometimes which class, these tagmata can appear different. Sometimes we have the fusion of different segments, and we will go over what their tagmosis looks like depending on the subphylum and class that we are talking about. All right, so the first subphylum Chelicerata. The organisms in the subphylum Chelicerata have two tagmata. So remember in this previous slide, there's 3 possible options, but there can be fusions of these tagmata. So for our subphylum Chelicerata, they have a fused head and thorax. This is called a cephalothorax. There's that word again, ceph for head, cepha. Like with our cephalopods, head, foot, this is a cephalothorax, a head and thorax. And then they also have the abdomen, which houses all of those internal organs. Some modified appendages that this subphylum have. They have modified mouth appendages. One of these is called chelicerae. The chelicerae are pincers or fangs, mouth parts for feeding, tearing, or secreting venom. We see that in spiders. Then we have pedipalps. Pedipalps are like tiny arms. They're used for capturing prey. They can sense the environment, and sometimes they can be used for copulation, so for sexual reproduction. And then they have 4 pairs of walking legs. This means they have 8 legs total. One thing that really separates them apart from things like insects is that they do not have antennae. So they're using those pedipalps to sense their environment, not their antennae. All right, the first class under the subphylum Chelicerata. So phylum Arthropoda, subphylum Chelicerata, class Merostomata. These are our horseshoe crabs. Horseshoe crabs are very, very old. They're a very ancient specimen in the fossil record that show that they've been around for an extremely long time. So they are an ancient semi-aquatic arthropod. There's currently only 4 species of horseshoe crabs still being able to be able to found in the world today. They're abundant in the Atlantic coast and the Gulf of Mexico. So these come onto the eastern shores of North America about once a year. They have a hardened outer shell. This is called the carapace. So the carapace is the shell covering, and it covers that cephalothorax. Again, they have 2 tagmata, the cephalothorax and the abdomen. And some special appendages that the class Merostomata have are book gills, so gills that are going to be used for gas exchange. The pedipalps are tiny arms near the mouth, so they're going to be located up in this region, right there. That's a pedipalp. And they have chelicerae. These are going to be used as pincers. And then finally, they do have another specialized appendage called the telson. I don't have it all the way here, but this tail, called the telson, stretches all the way out. And this is used to flip themselves back over if they get flipped over onto the round part of their shell. It's kind of that I have fallen and I cannot get up type of situation. They use that telson to help right themselves. I do want to, before we go on to all of the spiders, for anyone who is getting into the medical field, I know some of you in the class are going into medicine, you should really thank the class Merostomata. That is because the class Merostomata have a very, very special type of blood that is referred to as blue blood. Their blood is capable of detecting any kind of bacterial infection in a vast amount of space. So they would be able to, a drop of their blood could detect some kind of contaminant in the ecosystem if it was the size of like an Olympic-sized swimming pool. One drop of blood could detect a contaminant in that much water. So humans have benefited off of this contaminant detection capability of their blood, and we have since figured out how to harvest this blue blood. So we actually, human beings, collect horseshoe crabs every year, and we excrete, that's not the right word, we collect their blood in these giant vats, and it doesn't actually kill the horseshoe crabs. So we collect the crabs, collect their blood, and then release them back into their natural environments. We will then use that blood to create different types of contamination testing equipment and products, and they are heavily used in the medical field to detect contamination in medicine and on products and on equipment, etc. So thank your horseshoe crabs for the medical industry. Thank your horseshoe crabs. Okay, now we are going to see a lot of spiders. So be prepared. If you are an arachnophobia, just don't look and listen. Okay, subphylum Chelicerata, class Arachnida. These are spiders, scorpions, ticks, mites. So that's an example of a tick. Scorpions, different types of spiders, tarantulas. And this is just to kind of showcase how big their eyes can get. So this is a visually hunting spider. So the class Arachnida. In this class, scorpions are actually the oldest type of arachnid, and they are found on land. They have 4 pairs, so 8 legs total, and then 1 pair of pedipalps. The chelicerae are modified as venomous fangs, and then they also have this gland called spinnerets. The spinnerets are silk glands, and that's what the spiders use to form webs. Now, they do not have an excretory system like we have. Instead, they have this structure called the Malpighian tubules, and this is what's acting as their excretory system. So they will get rid of some waste through these Malpighian tubules, and they're essentially acting like their kidneys. I just wanted to throw up a few pictures of some amazing arachnids that I've personally gotten to see in the wild. So here on the left-hand side, this is a scorpion that I found in Panama. What's really amazing about scorpions is that if you shine a black light on them, they fluoresce, they glow this bright, bright color, and it's absolutely amazing to see in person. However, I never recommend shining a black light on the Arizona desert floor because you would be horrified at the amount of scorpions that are present in Arizona deserts. And that's probably true for anywhere in the southwestern deserts. This is a type of tarantula that we were never really able to fully ID, but it's a gorgeous example of a spider. We have this bright orange body and almost these bluish black legs. And then you can see that in this spider it has some really interesting growths from the abdomen, probably used to deter things from eating it, considering the bright coloration and the very pointy end. All right, next we are going to get into the subphylum Crustacea. So crustaceans, that should sound familiar. Crustaceans, this is crabs, lobsters, and if you recall from our Cnidaria lecture, I said the hydra eat these small crustaceans called Daphnia. Well, here's what a Daphnia actually looks like. It's a small crustacean, almost invisible to the naked eye. If you have some of your bottle experience, oh, you can't go into lab anymore. I bet in some of your bottles in lab, some of you might have these tiny Daphnias swimming around, but I guess you're not going to be able to see that, which is a little bit of a bummer, so that's all right. We will move on. All right, subphylum Crustacea. The class is Malacostraca. They can be found in marine or freshwater ecosystems. Their appendages are biramous, so they are double branched. Biramous, double branched. They also have these structures called swimmerets. They used in sperm transfer for the males and carrying eggs in females. So here are the swimmerets over here. And so males will, the eggs would be on the swimmerets and they will get fertilized and then they would be carried along on these swimmerets. They can have 2 or 3 tagmata, either a head, thorax, and abdomen, or a fused head and thorax, called that cephalothorax, and a segmented abdomen. So here's that segmentation. It's kind of put into different bars here. They have compound eyes, which is an eye with many lenses. They have gills for respiration, and these gills are actually also going to be attached to the legs. This can be really beneficial for those in the class Malacostraca, because if it's a swimming organism and its gills are on the leg, well then as it's swimming, moving those legs, it has more access to water, which means more access to oxygen, so they can essentially swim and breathe at the same time. Just like with our horseshoe crabs, they also have a hardened outer shell called the carapace. And I'm kind of going back up here to point 2, where I talked about their biramous appendage. What that means is that at the end of the appendage, it splits into 2. So if you look at this image here, we have our appendage, and then it splits into 2. And this is how all of the appendages would be for the class Malacostraca. All right, next we are getting into the subphylum Myriapoda. These are our centipedes and millipedes. They are terrestrial organisms, so they're on land. They have 2 or 3 tagmata. This is different than the other subphyla we talked about. Rather than a cephalothorax, they have a head and a segmented trunk, so it's a fused thorax and abdomen. They do have 1 pair of antennae, and for breathing, they have these structures called trachea and spiracles. This is for passive breathing, passive vessels for gas exchange. The spiracles are openings into the trachea. I do think we're going to get a little bit more detail when we get into our insects on spiracles and trachea, but essentially, along the body of the organism are small openings, small holes, that lead into the trachea. So the small openings, small holes are the spiracles, and then the trachea is inside. First class is for centipedes. This is the class Chilopoda. These are the ones that are not necessarily the most friendly. They can be highly venomous, especially this red-headed centipede. Don't worry, we don't have to worry about this one in Canada, I don't think. But they are venomous, they are predatory, and sometimes, no, I lost track of thought. Never mind, we will continue on. Okay, the class Chilopoda. They are going to live in the soil or under logs and stones. They are carnivorous, so they are actually hunting and taking down prey. They are active predators, and that's mostly going to be small invertebrates such as spiders. But I have seen evidence of a very large centipede taking down things like small reptiles. So they can be quite powerful. They have 2 tagmata, head and a segmented trunk. They have uniramous appendages, so they do not branch at the end. So if you look at this end of this appendage, there's no branch at the end, like with our crustaceans. They have some modified feeding appendages called forcipules. These are venomous fangs that they use to subdue their prey. And they do have 1 set of antennae. So here's that set of antennae right now. That's a sensory organ. They have ocelli. Ocelli are very simple eyes. Very simple eyes. These are mostly going to be used for sensing light. I have had personal introduction into their use of ocelli because sometimes they can be attracted to light. And when I was trumping around the forests of Panama, I had a headlamp on since we were looking for salamanders at night. and I turned my head, and my headlamp hit a centipede, and it was attracted to the light of my headlamp, and so it jumped toward the light, aka it jumped toward my face, and it was horrifying. Yeah, so ocelli, light sensing, simple eyes. And another characteristic of our class Chilopoda is that they are dorso-ventrally flattened. Dorso is for the dorsal side, the side with the back. Ventral is the ventral side, which is the side of the belly. So dorso-ventrally flattened. They are flattened on the back and on the front. So they're flat. Next, we have the class Diplopoda. Diplopoda are our millipedes. They are a little bit more friendly looking than our centipedes, and they're not going to be active predators. They are much more mellow when compared with their close cousin in the class Chilopoda. They are nocturnal herbivores, so they are not carnivores at all. They are going to be chowing down on detritus, which is like leaf litter, so leaves that are on the ground. They have two tagmata, that head and segmented trunk. They have uniramous appendages, so again, not split at the end, and one pair of antennae. They have cylindrical bodies, so their bodies are cylindrical rather than being dorsoventrally flattened. They have odoriferous glands. This is a gland that excretes, it can be toxic, very foul tasting, and it's a defense mechanism. If they feel threatened, then they can release some toxins and prevent themselves from being eaten. All right, next we have our last, I think this is the last subphylum for this part of the lecture, the subphylum Hexapoda. Our class for this subphylum is the class Insecta. So these are the first animals to fly. Most will have two pairs of wings on the thorax as adults. So here we have a bunch of different examples of everything you can find. Not everything, it's a massive, massive group of organisms. But these are just some examples of what you can find in the class Insecta. So praying mantises, beetles, stick bugs, butterflies, anything you can imagine. It's all going to be in the class Insecta. So there's between, obviously there's not one species, and I think that this number is probably a lot higher than this now. So 1 to 10 million different species, 70% of the kingdom Animalia. That is out of all of the sea jellies, the sponges, the mammals, the birds, the reptiles, and all of the arthropods. 70% of those are going to be some kind of insect. So a very large, diverse group. They have 6 legs, so 3 pairs total. Many of them can have wings. They have compound eyes and or ocelli. So here's a compound eye of our fly. And here's some ocelli. These are used again for sensing light. There's a lot of different types of specialized mouth parts for our insects. If you think about all the different types of insects and what they do, each one of those insects is going to have its own specialized type of feeding structure. Moths and butterflies are going to have those long coiled proboscis to get down to the nectar. Things like flies have a sponge-like mouth part to sop up the food of the substrates that it lands on. A mosquito is going to have a piercing-like mouth part to pierce the skin of its food source. So a lot of specialization of mouth parts. They have 1 pair of antennae, so 2 antennae total. And they do always exhibit 3 tagmata, head, thorax, and abdomen. And there's 2 different kinds of metamorphoses. So depending on the type of insect that we're talking about, they can undergo a different type of metamorphosis. First, we have incomplete metamorphosis. Incomplete metamorphosis consists of roughly 2 life cycle stages. They go from a nymph, and this is the term I want you to remember, a nymph, to an adult. The metamorphosis, the nymphs are going to be wingless, but look like their parents. So they look very similar to each other, just without wings. Wings are going to develop in pads along the back. So here we have some of those wing pads already coming out. And nymphs are going to undergo several molts. These molts are referred to as instars before reaching maturity. So they're not going to they're not going to go through what we think of a metamorphosis like with a butterfly. Rather, they're just going to keep getting larger. And as they get larger, they shed that exoskeleton. But it's not a real metamorphosis. They're just growing out of their own skin. So each time they shed, they go into a different instar. Because the juveniles and adults look so similar, it's safe to assume that they're also competing for very similar resources. So juveniles and adults are going to share the same resources for each one of these life stages. And the wings will come out on the final molt. So here's our final molt, and then here's our wings. Complete metamorphosis. This is the one that we probably think of when we think about butterflies. There's 3 distinct life cycle stages: the larva, the pupa or cocoon, and then the adult stage. So the larval stage is going to be that caterpillar. The pupal stage is going to be, well, the pupa. And then the adult stage would be the winged butterfly. So metamorphosis, the cocoon, the body of the larva will develop into the adult. There is an overall chemical breakdown of the larval body. So inside of this cocoon, the body of the caterpillar is chemically being broken down and then reshaped into a beautiful adult butterfly. So new cell formation and recombination is what helps the caterpillar go to a butterfly. There's really amazing YouTube videos of watching this chemical breakdown and then cell formation and recombination happen in sped up time. If you want to see something really cool, I highly recommend seeing something like that. So obviously, unlike our incomplete metamorphous organisms, our caterpillar looks quite different from our butterfly. And caterpillars are going to be eating things like leaves, where our adult butterfly is going to be looking for nectar to feed off of. So we have the prevention of resource competition between juveniles and adults. They eat different foods, and they're going to be living in different habitat. The caterpillar's stuck to living on its plant. The butterfly has the ability to fly away. So here's a cool GIF of showing this caterpillar going into its cocoon. Okay, so now we can go into some camouflage characteristics of insects. So camouflage is going to be used to either avoid predators or be a very good predator. So avoid predation or to capture prey. So here's a mimic mantis. So this mantis looks like this orchid flower. Mantises have all sorts of really amazing types of camouflages, and they are predatory. They are predators, so theirs is to capture prey. Leaf bugs, on the other hand, are going to be using theirs for camouflage, to blend in so that they don't get eaten. This is one of my favorite examples. This is a butterfly that looks like bird poop. So nothing wants to eat bird poop, so it's evolved to look like poop. Stick bugs blend very well in with their environment. So here's our stick bug here. And then you have ones that will mimic other predators. So this moth mimics what an owl looks like so that it can scare away potential predators. Here are some amazing insects I have personally been able to see in the wild. This is a mantis, a very small mantis that is the size of my fingernail, so very, very tiny. This is a beetle called a Hercules beetle, I believe. I could be wrong, but I think so. It was massive, absolutely huge. I actually have scars from where its little barbs on its leg. It wasn't trying to hurt me. It was just like, hanging on. And the barbs on its legs, while it was hanging on, left some scrapes and eventually turned into scars. So it was a very, very big beetle to mess around with. Beautiful moth examples with like these glassy wings. You could actually see straight through those wings. Here's another cool example of one of those cicadas. And then it was really, really awesome to see leafcutter ants. So we've talked about leafcutter ants a couple of times in class and how they make their little fungus gardens. So they're a really cool group of organism. All right, we are going to end here for now, and we will pick up again and go into, Echinodermata.